As I pointed out in the previous figure, a substantial amount of energy is released during the electron transfer process. This energy, released in discrete steps, is stored, as I said, in a proton concentration gradient, and the standard free energy available from these reactions is shown in this table. So as you see, when the electrons pass from NADH to Q, there is an amount of energy released equivalent to minus 70 kilojoule per mole, while there is no energy released with complex 2. In other words, complex 2 is not involved in the energy production, as you will come to see later. While in complex 3, there is again an energy of about minus 37 kilojoule per mole released, and so is the case with complex 4, minus 110 kilojoule per mole. So complexes 1, 3, and 4 are associated, as you will see later, with the creation of a proton gradient and with the synthesis of ATP, while complex 2 is not. Electrons flow through the electron transport chain components in the direction of increasing reduction potentials, where NADH will act as a strong reducing agent, and the terminal acceptor of these electrons, which is oxygen, is a strong oxidizing agent. And as I said in the previous slides, ubiquinone shuttles the electrons between complex 1 and the 3, or complex 2 and the 3, while cytochrome C shuttles the electrons between complex 3 and complex 4. Eventually, complex 4 will reduce oxygen to oxygen being the terminal electron acceptor. Ubiquinone, as you learned earlier, is a lipid-soluble molecule, okay, that accepts electrons, 2 electrons to be reduced to UH2, ubiquinol, and it can handle 1 electron at a time, as you learned earlier. Cytochrome C is a protein that is located on the outer surface of the inner mitochondrial membrane, and it shuttles the electrons, as I said, between complex 3 and complex 4. The sequence of electron carriers in the respiratory chain can be demonstrated by using inhibitors that bind to specific components of the electron transport chain. Here, for example, you see that rotenone is an inhibitor that can bind to complex 1. What does that mean? When rotenone binds to complex 1, it will block the transfer of electrons from complex 1 to ubiquinone. Therefore, complex 1 will remain reduced while complex 3 and 4 will be oxidized. There are inhibitors for other complexes, such as antimycin A that inhibits complex 3, cyanide that inhibits complex 1, malonate that inhibits complex 2, as you learned earlier, which acts as a competitive inhibitor. So using these inhibitors, we can determine the sequence of these carriers, and we know then that the electrons will pass from complex 1 to 3 to 4, or from complex 2 to 3 to 4. Now I'll start discussing these complexes. Complex 1, the systematic name is NADH ubiquinone oxidoreductase, because it transfers the electrons from NADH to ubiquinone, reducing it to ubiquinol, as we learned earlier. In this complex, there are electron carriers, such as FMN. There is at least 8 iron sulfur clusters. How do the electrons pass from complex 1 to complex 3? This figure shows the transfer of electrons from complex 1 to ubiquinone, so that ubiquinone can deliver them to complex 3. NADH will lose 2 electrons as hydride ion, and these will be used to reduce FMN to FMNH2, and then FMNH2 will lose the electrons one at a time to iron sulfur clusters. There are like 8 iron sulfur clusters in this complex, and the iron sulfur clusters can deliver the electrons to quinone one at a time. You recall that quinone, when receiving 1 electron, will form the stable free radical semiquinone, and then when it receives another electron with 2 proton ions, it will be reduced to QH2. So here, The pathway of the electrons starts from NADH through FMN, through iron sulfur clusters to reduce Q to QH2, which will shuttle the electrons to complex 3. As the electrons pass from NADH to QH2, they will lose some of their energy. The level of the energy in QH2 is less. That's to say NADH has a more negative oxidation reduction potential. So what happens? This energy will be used to pump protons from the inside of the inner mitochondrial membrane, from the matrix side to the intermembrane space side or the outside. And every 2 electrons passing in the system will be accompanied by pumping 4 protons to the other side of the membrane. It's believed that there is a proton pump, which is possibly H plus and A plus antiport pump that is located in this membrane, and that results in pumping of these protons to the other side of the membrane. The second complex, complex 2, is also named succinate ubiquinone oxidoreductase because it accepts the electrons from succinate, and through several electron carriers in the complex, it will catalyze the reduction of ubiquinone to ubiquinol. It is also called succinate dehydrogenase complex. As you recall, this complex is part of the enzymes involved in the Krebs cycle. And if you recall, at that time, I said all the enzymes in the citric acid cycle are located in the matrix, with the exception of the enzyme succinate dehydrogenase complex, which is located in the inner membrane of the mitochondrion. This complex contains 3 identical multi-subunit enzymes, making what we call a trimeric structure, and the overall shape, as you see in the next slide, is a mushroom, where it is made up of a head and a stalk. The stalk is made up of 2 subunits, 1 or 2 subunits, while the head is made up of 2 subunits or 2 polypeptides. As you see in this figure, the electron carriers are present in the head part and the stalk part of the complex. In the head part, you have the substrate binding site, which is the site where succinate will bind. There is FAB, and there is at least 3 iron sulfur clusters. While in the stalk, there is the ubiquinone binding site and a cytochrome B. How electrons pass through this carrier? When succinate is oxidized to fumarate, the 2 electrons will be used to reduce FAD to FADH2. FADH2 will release these electrons one at a time to reduce the iron sulfur clusters. And the iron sulfur clusters, again, will reduce the Q to QH2 one electron at a time. It seems that cytochrome B has no role in the electron transfer in this complex. Another thing that you should remember when studying this complex is that this complex is not, when the electrons pass through it, it's not accompanied by pumping protons to the other side of the membrane. In other words, this complex is not associated with the production of the energy ATP, as you will come to learn later. This slide shows a single copy of the complex 2, or the succinate dehydrogenase complex, showing the electron carriers, as I said, FAD, FAS clusters, the 3 of them, as well as cytochrome B. But as I said in the previous slide, cytochrome B is not involved in the electron transport in this complex. Now I come to discuss complex 3. Its systematic name is ubiquinol cytochrome C oxidoreductase, because it takes the electrons from QH2 and they will pass through this complex, deliver them to cytochrome C, which will shuttle them to complex 4. Now, the oxidation of one molecule of QH2 in this complex is accompanied by the translocation of 4 protons across the inner mitochondrial membrane. So this complex, as you will see, is involved in the production of energy, and these 4 protons, 2 of them come from the matrix and 2 of them come from